So it's my pleasure to introduce Farzana Nasrin, who just joined us this year. She graduated from Texas Tech with a PhD in applied mathematics in August of 2018. And her research interests spanned algebraic topology, differential geometry, I think I said that right, statistics and machine learning. She's currently an assistant professor here at Manoa in the Department of Mathematics. And before coming here, she was working as a postdoctoral research associate funded by the, I don't know what the ARO is.
Army Research Office.
Okay, Army Research Office in Mathematical Data Science. So she's been working on building novel learning tools that rely on shape peculiarities of data with applications to biology, material science, neuroscience, that's my favorite, and ophthalmology. Her dissertation involves the development of analytical tools for smooth shape reconstruction from noisy data and visualization tools for utilizing information from advanced imaging devices. And the title of her seminar is Bayesian Topological Learning for Complex Data Analysis. And I will turn it over to you. Welcome.
Thank you. Let me share my screen. Yeah, thank you for inviting me. This is really interesting and it's good to see so many people, at least virtually. So this is, I hope you can you see the screen, my slides? Okay, I just need to make it full screen. 1 second. Somehow I don't see the one. Oh yeah, I found it. Okay. Okay. So I mean, I didn't actually know that neuroscience is your favorite. Then I would probably add that as our application for today. But today I will be explaining something from biology. The title of the talk is Bayesian Topological Learning for Complex Data Analysis, but I will go kind of in the reverse direction. So first, I will explain what does it mean by complex data and what type of data we can use for like analyzing this thing, and probably our method will be applicable for those type of data also. So I will explain about this a little bit, and then I will introduce that why topology sounds like a good fit for analyzing this type of complex data, because people are trying to analyze this type of complex data with many different methods, and suddenly for like 10 to 15 years, there is a big jump of working with topological data analysis. So this field is really booming and it's really interesting. So I hope there are several students. So I will introduce this field with you guys. And finally, I will talk about like a Bayesian novel approach we introduce in our lab. And I will show you a classification problem from biology. Okay, so before starting, it is just an acknowledgement I want to do. Like I worked on this project with a graduate student and she recently actually graduated and joined industry. So Cassie Mikuchi and Basilis Marlos was my mentor, my postdoctoral mentor at UTK. And we worked with the biochemistry department and chemical engineering department. So they had this interesting problem and they were trying to solve it for many days. They had different aspects of the problem. And one time they actually shared this problem with us, and we found a nice, interesting solution of that. So the problem or the type of data set we are actually working on, it relates to plant cell, and plant cell has a nice phenomena. It's called cytoplasmic streaming. So cytoplasmic streaming is actually a simple flow inside of plant cell. So usually those flows are like liquid items. So they carry these items from one place to another. But this cytoplasmic streaming is very, very important for at least having normal growth of the plants. And that's why this full movement should be organized. So how we can organize this movement or intercellular movement inside of plant cell? So that the structure which mainly does that is called cytoskeleton, is like a skeleton but for the plant cell. And this cytoskeleton actually determines the structure of the cell, and also it involves the cellular motion. So what it does that with this cytoskeleton, they have several filaments. So these liquid items, they actually attach to the filament and then they move towards these filaments. There are several research done already by knowing that how these flows are doing. But one question is still remain unanswered, is that what are the main reasons behind having this structure? What is the biophysical mechanism which are responsible for having that particular structure, or why that is involved with the motion inside of the plant cell? So this biophysical mechanism, to understand the biophysical mechanism, what that research group is doing from chemistry and chemical engineering department, what they're trying to figure out, that they actually find out several measurements and several aspects. They feel like probably that is impacting the flow, and they want to see whether these hypotheses, what they are making about these items, whether they are correct or not. So then they contacted us and they said, okay, we have this actin filaments. So here you can see an image of an actin filaments, and you can see some of the places are like very bright and some of them are like very gray, very gray. So if I distinguish it into three different regions, green, yellow and blue. So if you look at the green portion, you can see like several thicker place where the light is like very bright. And if you look at the blue and the yellow part, it seems like they are mostly gray. So if you think about this as like road map, probably the green will be the analogous to the like highways. And this gray one, probably this where the light is not very prominent, that can be like the local roads, like Dole Street, where I live. So now, if we want to actually characterize it, how we can do that? So they gave us that, they actually informed us that they are thinking about the cross-linking protein number. So what does it mean by the cross-linking protein number is like when two filaments actually touch each other, they join at several places. So how many places they are actually making this connection, that's called cross-linking protein number. So they find that the cross-linking protein number, or they make a hypothesis that is probably impacting these shapes. So they gave us like different simulated data where they have different type of cross-linking protein number. So for example, if you look at the first black images, those black filaments, they are actually with cross-linking protein number 825. The next one, the rates are 1650, and the last one is 3300. So when they showed this figure to us, like they are trying to classify this, whether there is any method which can classify them properly. And since we are mathematician, we are thinking about it like, okay, yeah, it's possible. The most prominent thing we can see from this one is like, you can see some loops forming for the green and the red one. So if you look at the green or the red one, you can see some very prominent loop. Whereas if you look at the black one, it seems like there are some small roads which are always crossing the path. So it is not forming a bigger loop, whereas in green and blue, it is forming a bigger loop. So we said, okay, let's think about which part of mathematics deal with holes. And that's why we find that topology can be a good fit for this one. And I will explain it more why we feel like topology will be a good fit. So the thing I wanted to introduce today is called topological data analysis. It's a very promising field for data analysis. And the whole motivation was like thinking this, that every single data has a shape inside of it. Maybe we don't see it, but it has a shape. And we want to we actually want to find out the shape information from the data. That is the that is the motivation, and definitely you can understand that it will be using techniques from topology, which is a field in mathematics for like 250 years. But recently, for like 10 or 15 years, people are using it for data analysis. So that's what I was saying. The motivation is like data has shape, and shape is very important. It can give us a good insight of the data. So rather than looking at the topology, let's see what does it mean by shape. Data has a shape. So here I'm giving you like 3 examples. If you look at the first one, this is very popular. We all know about this. It's the linear type of data, and we do linear regression with that. So we know that this data set, if we want to do some machine learning algorithm like linear regression, then we know that we can actually fit a straight line. So that means this data set has a shape which is a straight line. And if we can fit that, we can actually extract lots of information from them. We can say that, okay, one variable is related to other variable, how they are related, whether they are proportional or inversely proportional, or many other information. So that means once you know the shape of the data, you can actually talk more about the data. In the second one, we can see several clusters. Rather than looking at the clusters, we can probably think about, okay, there are 4 nodes, and we have to consider it like this. Or it can be a circular shape. Those who work with signals, they know that circular shapes are really important. It relates to periodicity. So from all of this, we can see that there are some intrinsic shape underlying of this data. And probably the first thing which will come to our mind that, okay, maybe we can use geometric modeling. The problem with geometric modeling is it's very rigid. You can actually have only some type of shape, which cannot probably incorporate the wide varieties of data we can actually encounter for our real life. So that's why we go for topological modeling, because topology is very flexible, and we will see how. Okay, so I will talk about a joke. So there is a joke about topologist. So who is a topologist? The answer is someone who cannot distinguish between a donut and a coffee cup. I guess some of you probably heard about this thing. So just to show you that what does it mean by they cannot distinguish, I just want to show you the video. So just a second. So if you look at this coffee cup carefully, you can see it seems like it is kind of filling up, and then suddenly it becomes a donut. So what it is doing that, I want to show you, oops, sorry. I want to show you one more time. Yeah. So what it is doing that it can do some type of deformation. It can do stretching, it can do bending, and it can preserve some properties throughout the deformation. So this was the main motivation of using topology, because topology can deal with deformation, and this deformation are something we always see in real data. Real data are noisy. They can incorporate noise, measurement noise, or several other type of noise. So topology can be really useful because it doesn't matter whether you are deforming it, it can identify it. So that was the main motivation about using topology. So, but like, let's talk about a simple problem, and probably many of you are familiar with this problem, but that was considered as the first applied problem for topology. Is the 7 bridges of Königsberg. How many of you know this problem? None of you? Okay, so this is probably from 17th century. So this is a kind of tourism problem where it has like 7 bridges, and the question was whether it's possible that if you stand on one bridge, whether you can cross all of the bridges and come back to the initial place, but you have to cross the bridges only once. So crossing all of the bridges only once, whether it's possible or not, that was the question. And it was actually investigated by Leonhard Euler. So he's a very famous mathematician. So he solved this problem in a totally different way. He thought, okay, it doesn't matter how wide the river is. It doesn't matter how long the bridge is. The only important information is there are some land masses and there are some bridges which are connecting them. So rather than looking at this complicated figure, he just transferred this full information into a simple graph. And you can see that on the right side. So he said, okay, let's draw this, let's actually have this connection by some edges, and the nodes will be those land masses. And from this figure, he actually found several other formula, and he solved this problem. He actually found a really, really nice one-line algorithm which will solve this problem. I'm not going to share this with you. This is kind of your task. Right after this talk, you can Google for it. It has a really nice solution. You cannot actually do that. So it is not possible to cross all of those bridges by just going once. Okay, but you should try this after the class if you are not, after the seminar, if you are not familiar with the problem. Okay, so that was kind of a motivation that from a data set, we are going to build a structure. Okay, so how we are going to build a structure that will be giving us by using these simplicial complexes. So simplicial complex is like a collection of simple pieces. For example, it can be a point, it can be a line. So if it is a point, we call it 0 simplex. If it is a line, we call it 1 simplex. And then triangle, 2 simplex, and so on. So we are going to use these simplices to build a structure from a data set. And we will see, I will show you how. So that's one information needed when we are going to build the structure. Another information needed is called homology. Homology is actually, again, like a concept from topology, which actually analyzes the whole of a subject. So whole of an object. So whole means like a 0-dimensional whole. It's actually a point, a connected component. 1-dimensional whole is like a loop. 2-dimensional whole is like a void, like a volleyball, which inside is totally hollow. So this homological information, that 0-dimensional homology or 1-dimensional homology, can actually help us to summarize a very complex data into just a simple graph. And that's the main motivation of using topology to analyze data. So what we are going to do, we will build a shape on top of a data set, and we will see whether that converts to something we know already. So this is like a kind of topological data analysis 101. So we will just learn we are going to learn the basic way of finding from a very simple point cloud, how can we build shape characteristics and summarize it in a figure or a diagram, and we call it persistence diagram. And I will explain why the name is persistence diagram. But the idea is, if you look at figure A, here we can see like 7 points. Okay, so remember, points are 0-dimensional homology, so these are connected components. So when you have connection, these are connected components. So what we are going to do, we are going to build a circle on top of it with a smaller radius. So we do that and you can see this in B. So still we can see there is no connection between any of them. So we still have 7 connected components. We increase it a more, and now it seems like the top 2 are connecting, the middle 4 are connecting, and the last one is separated. So can anyone tell me, if you look at figure D, how many connection we see, how many connected components we have now? D. Okay. 3. It seems like I'm teaching. I do that for my class too. Yeah, thank you. That's the right answer. So from 7 connected components, now we move to 3 connected components because we make some connection. So we will continue this more and more. And after a certain time, remember what we are trying to do is that we want to see that whether it can converge to some other shapes. So we have 0-dimensional homological, which are connected components, then 1-dimensional homology can be loops. So if we increase it more here in E, we can see that there are two connection making it this leftmost points, rightmost points. So it seems like there is a loop forming. So here we see that not only we are making connection, we are also forming a loop, and we call it 1-dimensional homology. So from our data, now we can see a structure is building, and the structure is probably a loop with a string. So rather than actually taking all of this information, we can actually summarize it as a persistence diagram, where you can see those black dots, all of the black dots. So x-axis will be a birth and y-axis will be a death. What does it mean by a birth and death? So birth means when you first see those homological features. So remember at the beginning, we saw 7 connected components. So that's the starting of the homological feature. And we say that's the birth point of 7 homological feature. So then we keep continuing this figure, we keep continuing increasing the radii, and we have those merges. So here it should be like 7 black dots. You cannot see all 7 because there are some overlaps. But these 7 black dots are saying when the first one was connected, the second one was connected, and going on. The red triangle is the place when we first saw the loop is forming. So that is the birth point of the loop, and we will continue the process until we get to that point where everything merged to one connected components. So you can actually get this from this figure. So if we keep increasing it, what will happen? So everything will be merging to one place. So that will be the last connected component, and we will stop our process. So see, from this data set, now we can actually have a nice, compact and informative way of presenting the data set. Maybe you can say, okay, the data set was not complex because I was trying to actually teach you how to do this persistence diagram. But I will show you what type of information we can have by doing this topological summaries. It can be really, really useful, and it can be very informative. This is just a simple thing, like sometimes people use the birth and death, and sometimes rather than using the death, there is another way of presenting it. It's called persistence, which is nothing but death minus birth. So rather than looking at when it is dying, we just look at how long it persists. So it will just give us a nice flipping of this. It will just transfer the graph, like transform the graph a little bit, nothing else. It's just another representation of doing persistence diagram. So this is how we do the summary. Now the important thing of these summaries are they are not on a vector space. So I don't want to go too much details about this one because that will probably take us to too much mathematics, and I don't want to do that. I want to show you the basic idea. So the first problem we had that most of the statistical analysis or like machine learning algorithm, they actually build on a vector space or a Hilbert space. But the problem is the topological, the persistence diagram are not on a vector space. So that was the first problem we faced, and how we can deal with that, that is important. So you cannot actually use any simple Bayesian or any simple statistical analysis for doing that. There are 2 directions people are actually pursuing. One direction is rather than looking at the persistence diagram, they map it to a vector space or a Hilbert space. That one solution. And that direction is also pretty huge. Many people are actually working on that direction. There is another direction that you don't do that. In that way, if you are not mapping to a Hilbert space, you bring some more complexity. And now you are on that space, which is not a Hilbert space, which are just simple set, and how you can deal with that. So what I'm interested in is the second one. I don't want to map it to another space. I want to keep it in the persistence diagram space, and I want to see whether it can solve the problem of finding densities. So the solution we actually found is called point process. So point process is an interesting phenomena in statistics. It's actually kind of coming from like a string from finite set statistics, but the whole idea, which was really promising for persistence diagram, is that when you do a persistence diagram, so let's get back to this one. When you do a persistence diagram, there are two types of uncertainty we can face. One, the number of points in a persistence diagram can vary. And if you just incorporate some amount of noise, this number of points can vary a lot. So we have to incorporate that information that the number of points is a variable. That's first uncertainty we have to address. The second one is how the points are distributed on the space. So here it seems like the domain is on the first quadrant, how the points are distributed. We have to actually find a density for that. So that's the whole idea, and point process in a constructive way actually does that. It actually defines a set of points probabilistically. So you have a scattered point, scattering everywhere, but it will define the density of it, and it will also incorporate the information that you can vary the number of points. So this is an information we are going to use, but remember we said this one is about Bayesian. So I'm actually giving you like all of the building blocks, and then we can see that the main algorithm. So Bayesian statistics was like very promising because for the first time we thought about, okay, it is not only depending on the samples, we can actually incorporate some expert opinion, some data, some historical information. For example, if we think about, like I did a neuroscience application where we are actually giving our some baseball player like to play a video game. Some of them are expert professional baseball player, and some of them are like amateur, they are just playing the video game. So, and when they're playing the video game, we are collecting their EEG data. So it's like a brain signals. So we are seeing that when they are actually hitting the ball when they should or they should not. And we calling this application as like, or this approaches as like go or not go. So, and we are seeing that when you are taking a wrong decision, especially if you are a professional player, but you are taking a wrong decision, how your brain is impacting it. So the signals are different. And suppose you are taking a right decision, what type of signal it is generating. So we can actually incorporate this information that probably some of them has an expert knowledge of playing baseball or some of them don't. We can actually do that by using Bayesian. So that's why Bayesian is really useful. The idea of Bayesian is that once you have some prior belief, so you have some prior information about an event, and now you collect data. So you want to see that when you collect the data, whether it changes your belief or you actually think about, okay, whatever your prior belief is, it is actually finding a similar analogy. So that's how Bayesian works. So it wants to update your prior information or your prior knowledge in the light of the evidence. So that's how we do Bayesian. So let's see about Bayesian for a persistence diagram. So if you look at the first figure, which looks like a dumbbell or like a peanut. So if you look at that, it seems like it has two holes, right? So it has two holes. So if we draw the persistence diagram, we know we have red triangles for holes. So it will just generate two holes. Now this is my prior information. Some expert told me that it should be a peanut shape. Okay. Now, suppose you are collecting the data and you did the worst thing possible. So the data collection was totally noisy. So somehow you don't see the peanut anymore. It looks like an oval. So it's so noisy that this peanut shape is kind of hiding behind this oval shape. So that means the person who is collecting the data or collecting the evidence is not doing a very good job. But can we actually extract the prior information? Is it possible to say that no, it should be a peanut shape from my prior information, even if I have this kind of noisy data? So that's what Bayesian does for a persistence diagram. So persistence diagram will make it a compact summary. So it's even more challenging. We are not actually looking at the data. Now we are looking at the compact summary. So if you look at the compact summary, it seems like this one point is rising up. So it's saying that maybe there is just one loop. And when you have points close to the vertexes, we mostly feel like they are noisy because they just born and die, born and die, born and die. So probably like noise. So that's why we mostly discard them, or we feel like these are not important information. So it's a very bad news that from 2, we just have one loop now. So can we extract that information? So that's what is done by Bayesian in persistence diagrams. So this is this is the foundation of it, like how we form the Bayesian, what type of point process we can actually incorporate. And we actually did several point processes already, but the initialization was with Poisson point process because this is very popular one, and it just needs one parameter to consider, which is called intensity. But again, I'm not going to do too deep on this one. Here, the reference of the paper, which is already published in SIAM Journal of Data Science, Mathematics of Data Science. So if you are interested, you can look at it. It has the full information about how you can build the Bayesian, depending on a point process theory. And how you can do the likelihood, that's actually related to a mark point process. So mark point process is just like once you have one point, you think about it should match with someone or some other data. That's called marking. And you probably have an idea, okay, marking means like, okay, now I am actually attaching my prior belief with my evidence. So that's what mark point process is going to do. So this is just an analogy between the regular Bayesian and with the Bayesian for persistence diagram. I wanted to show you this one because in a regular Bayesian, we always look at the data. Whereas in this persistence diagram, we are looking at a persistence diagram, which is not a data. And it is not even like a vector. It is actually a set. So even after doing this thing, we find a nice analogy between Bayesian, like what we usually expect from a Bayesian, we can see it here. And I will show you in next few slides that how we actually have the regular idea what we have for Bayesian with this event. Okay, so here, just think about you have two persistence diagram. The black squares are your prior information, like what you already know about your data set, and you collected the data, and dy or those red hexagons, these are your information or like evidence. So here they are showing that what type of matching we are doing. The idea was just to show you that it's not like we are depending on any specific type of matching, like, okay, if one point is very close to other, then we are just matching it. We are not actually doing that. Our algorithm is flexible enough to consider all possible mappings. So that's why I'm showing two different mappings possible. And these are all actually presented in the paper. But that's the whole idea about persistence diagram, because here you have sets. So you need to actually match those points. So this is the formula of Bayesian. If you are familiar with Bayesian, the Bayesian posterior, which is the final answer, it depends on, it actually has a formula that prior times likelihood. So if you look at this gray portion, it's actually likelihood times prior divided by the evidence, the integration, or we call it normalizing constant, which depends on the evidence only. And very interestingly that, yeah, it is a complicated formula, but it is creating an analogy with the regular Bayesian. So here is just how to find like a more applicable way of doing that. So we use Gaussian to actually find a nice application. I wanted to just show you how we can actually have a nice analogy with standard Bayesian for random variables. So here on the first figure, you can see a circle. So by circle, we see that, okay, that means we have already know what does it mean by Bayesian, what does it mean by persistence diagram, and it should give us 1 point. Okay, so by informative prior, I'm saying that I know the place of the point, and since it's informative, I have a very good understanding of my knowledge. So I put a smaller variance. So that's why you can see it's very tight prior. But if we use our data set, we have this posterior, where you can see this green dot. This green dot is actually coming from the evidence, but the posterior is almost same as prior with little bit higher variance. So it is saying that what we usually think about is that if our data is not noisy, if my prior information is very correct, I should get the perfect posterior. And we can see it here. Okay, now you can say, okay, you did a cheating, you did both of you actually take both of them as like nice prior and nice evidence. Okay, let's make it worse. So if we have even a bad prior, like weakly informative prior, we can have a posterior, but it can have a more noise or higher variance. Okay, so these are all showing that, but one interesting thing we found that if we change the parameter of the likelihood, that means if we trust more to our data, we can significantly improve our posterior. So here we can see that from the posterior, which was done by using the data set without any parameter choice, and then we make a nice parameter choice and we have a very tight variance. So that means if we just put a higher weight for my likelihood or for my evidence, we can actually improve the posterior. So these are all figures which are showing that for a regular Bayesian, what we expect, we can actually see it for Bayesian of persistence diagrams also. So let's see. Okay, so these are all like other figures, and we actually coded it. There is our package. You can go to GitHub and download it, where you can just incorporate, you can just put your own data set and some prior information. It will compute the posterior, and it can also do classification problems. So by classification, what we are trying to do, we are using a base factor classification. It's obvious because we are doing a posterior. So the base factor classification is nothing but ratio of the posterior. So which one has a higher weight, it will go to that class. Just to show you a flow chart, if you remember, these were the three classes we first had with the actin filament, with a low number of protein or higher number of protein, and we can actually find their persistence diagram. If you look at the persistence diagram, you actually find something very interesting, that this black one, when you do not have very prominent or bigger loops, the maximum persistence of a loop is going somewhere like 3 or 3.5. But when we had like this red or green, that means it will persist for longer. You have bigger loops. So here it seems like it is going up to 6 or more than that. This is just one example, but we found like it can go up to 6 or 7 because it has larger loop. And then we find the densities by using the Bayesian, and we divide the posterior. And if we get a higher values, we just assign it to that class. So that's how we do this one. So just to give you the result we got for this actin filament classification, and this is something I need to actually say, that there is no method yet for doing this filament classification. So we didn't have actually any comparable method to do. So we actually compare it with several topological methods or non-topological method. And we actually took 50 diagrams from each class. You may say like, why 50? That's a low number. But taking those simulation, it is very lengthy process. And once we heard that in one full week, they can produce maximum 25 images. So, and we are using like 150 of them. So it's like a very lengthy process. So that's why we had to actually use only 50 diagrams from each class. And we use a tenfold cross validation for doing the classification. So here is just a summary of our result. So our framework was giving like very high accuracy. Here I am giving the AUC, area under the curve. So you probably know that if it is close to 1, that means it's very good classification. So we found that our method can actually give us like more than almost like 93%. And there are several other methods we tried, like persistent images, persistence landscape. They are actually other topological method. Because they are on a Hilbert space, you can actually use many machine learning algorithms. So we use random forest, support vector machine, neural net, all of this, and we found like we even tuned them as much as possible. But it seems like our classification or several other topological classification is actually doing a better job, because those classes, those actin filaments, they have some topological information. And it seems like it was useful for finding or doing the classification. So here is the link of the actin filament classification paper. It's in archive. So if you are interested about knowing the application, you can look at it. So that's actually all for today. And thank you. Now you can ask me a question. Thank you all.
Hey, thank you very much. What a great seminar. So if folks could type their questions into the chat, and then I'm sure you'd be delighted to take questions. And you can stop sharing your screen, and then if you're asking a question, turn your camera on so she can meet you. In fact, everybody should turn their camera on, because Farzana is brand new to our university. She hasn't met anybody in person yet, so please say hello in person. It's one of those fun things about, you know, taking your faculty position in a pandemic, right? Just a little extra fun.
Yeah.
Okay. Any questions from folks? Let's see.
Maybe I can ask one question. I have one question and one comment. And can you use your technique for time series analysis, such as COVID data? That's my question. And then my comment is that your bridge problem, I think the diagram and the bridge, the ABC notation would be a little bit off. So you may want to double check it.
Okay. ABC, sorry, can you?
The Königsberg the bridge that.
Oh, okay. Okay.
Yeah, that one is kind of minor thingy, but yeah, I just looked through and it looked a little bit different. But anyhow, that's minor. But how do you use the your technique for time series analysis?
Actually, I didn't work with time series analysis at all, but there are several papers where they actually work with time series analysis. So one way of doing with time series is like, since they are signals, these are not point clouds, right? So the method I just showed, where you construct several like circles of different radii to see the shape of the data, you cannot do this for now. So there are two ways to solve it. One is by changing this time series or the signals to some point clouds, and we can do this by using the Takens algorithm. Are you familiar with that? Where you actually, okay.
Not really.
Oh, okay. So Takens algorithm is actually, so if you just think about a sine curve, okay. So they actually try to find out a point where you see the repetition. And rather than looking at the signal, they actually look at the repetition and they form like So a sine curve where you see a regular repetition, it will form like a circle. So it will just put some points on a circle.
Yeah.
So that's one way. First you take the signal, convert it to point cloud, and then find the persistence diagram. That's one way, but there is another efficient way, which is called sublevel set. Sublevel set is very useful. What you do that if you have a signal, what you do is like pulling your curtain up. So if you have this place where you can see some point or like you just pull your curtain up and you see how many connection it is forming, whether it is making one point and it is merging to another wave. Taking all of this information and all of this emergence, you actually categorize in a persistence diagram. That's actually a different type of persistence diagram. I didn't show you that, but sublevel set is very useful for time series analysis. Yeah.
Okay, thanks so much.
Here.
See. Other questions from the group? How about from one of, okay.
I
Will you
see
just
someone.
answer your question and say hi at the same time?
Yeah, I had a question.
There you are.
Yeah.
So I do a lot of work in human in the loop machine learning and finding like compressed representations of high dimensional data that capture structure in interesting ways. And I was just wondering if you're familiar with the UMAP algorithm. I think it utilizes like a very similar set of theoretical tools, like constructing cyclical complexes of the data in high dimensions.
Can you tell me what is the abbreviation of UMAP?
So it stands for Uniform Manifold Approximation and Projection.
Oh, okay, manifold approximation. Yeah, I hear about it. And this is something like in topology, there is another approach which is called mapper. Are you familiar with mapper?
I've heard of it, yeah.
Yeah, mapper is something like that. So it takes very high dimensional data and it just tries to find some overlap on the data. And for every single overlap, it actually create one nodes. It actually, so what it first, so suppose you have a circle or something, circular type of data. So it first takes like several discretization of the data and it put one node for each of them. And then if you have overlap, it makes a connection between them. So by doing this, from a very high dimensional data, you can find a very lower dimensional representation of that. That's very close to manifold approximation type of thing. Yeah, but I didn't actually, I mean, I heard about manifold approximation as a dimensionality reduction algorithm, but I never used it. Yeah. Does that answer your question?
Yeah, I guess what I'm wondering is, I guess that's trying to do something very different than what you're doing with the persistence diagrams, where you're just trying to identify like particular structural facets of the high dimensional distribution, right? Like, is there a hole or how many holes, that kind of thing, right?
Yeah, mapper is totally different. Well, I am working on mapper for a different type of data. It's actually a genomics data, RNA-seq data. So that's very high dimensional data with lots of patients. They either have tumor or they are healthy, and we are actually, and lots of gene expression, and we are actually using mapper for that. So it's not actually, we don't look at any structure behind the data. We just want to see that whether it can characterize healthy and sick, or even it can characterize sick because they have tumor or probably they have cancer. They should not characterize all of them in one place because they may have different genes which are responsible for that. So whether they can characterize them. So mapper is a totally different thing. Yeah, you are right.
Got it. Okay, thank you.
Yeah.
All right, any other questions from folks? Looking through questions. Okay, I'm not seeing any. This was a wonderful seminar. I think people really enjoyed it. Any last thoughts before we take off for our wonderful weekend where we're all going to stay inside and not go to the beach. Hi, Daniel Ford. I see you back there. How you doing? Okay. Well, thank you so much. And make sure you sign up for our I think we've got you on our mailing list, and we'd love to see you back here at the Fellows program at the end of the semester, and also attend our seminars anytime. And we all look forward to meeting you actually in real life, in person.
Yeah, I'm looking forward to it. I don't know when this will, I mean, for me, it's totally different. Yeah, I mean, moving to a new place and then you are stuck at home, you cannot meet with your colleagues. And I miss the thing that if you have a question, you can just go to your next door and ask, hey, do you know the answer of this? Now I always have to send emails and wait for the answer. So yeah, I wish this, I mean, I don't know, probably for this semester we cannot have in-person meetings. Hopefully next semester.
Doing our best right now. But we're looking to the future for sure.
Yeah. But yeah, thank you for inviting me. It was really interesting talking to you guys.
You're welcome.
Can I
Everybody,
have information?
sorry, any last thoughts? I think I didn't hear that last one.
Hello. Oh, hi. I'm Amihan. I'm just wondering if I could get your contact information.
Oh yeah, sure.
Yeah, like email.
Okay,
Yeah, your talk was very interesting. But I'd like to look into your papers further.
yeah, this is my, I just write it in the chat. This is my email, and let me share my website with you so that if you want to read some of my papers, that will be helpful.
Okay, yes, thank you.
Yeah, I mean, also you can just, okay. Yeah, this is my website. So I usually update it as much as possible. So yeah, definitely. And I mean, if any of you are interested about using the software we are building, the R package, it is still in the beta version. So we are accepting any type of feedback, comment, or if you can figure any bug, that will be really helpful.
All right, well, thank you so much again, and thanks for everybody for attending. I've got a great crowd this time. Really such a pleasure to meet you, and sorry, enjoy your weekend at home.
Yeah.
Okay.
Okay,
Take care.
bye-bye.
Aloha.
Bye.